Okay, so today is our 18th lecture, and we are closing the module on system biology. So now we are today we're going to talk about how we draw diagrams to connect entities like gene and protein in biology into networks, so that we can analyze them and visualize them. So today's agenda is simple. We want to look at networks type data, both in biology and maybe some example from real world. And we look at the properties that they have and some application that we can make. So I think we already saw several examples of networks in this course already. So networks in its simplest form, you only need two things. You need the nodes or vertices, and you need the edges to form a network. But in real life, the networks can be more complicated. You can have edges that are not directed, undirected edge. So this means that these two nodes are related in some ways, but there is no direction. But some edges, like this one, has an arrow, so that has a direction. So for example, when you have a transcription factor binds and regulate another gene, it's a directed interaction because the transcription factor acts on the gene, not the other way around. But if you have two proteins that can bind to each other, the interaction is both ways. If A can bind to B, then B can bind to A. So in the networks, you can have both undirected and directed edges. And you can have more than one type of edges. You might have a gene co-expression, two genes that whose transcriptomic profile correlate with each other as one type. But you might have the protein product for those genes being able to bind to each other. That is another type of interaction. So in several networks, you will see multiple types of arrows, multiple types of edges as well. And for each node and for each edge, you can also have attribute. What they call attribute is basically the data about the nodes and about the edge. So for the node, for a gene or for a protein, you might have the pathway annotation, you may have the expression level for that gene, for that protein. And for the edges, you might have the binding strength between two proteins, or there might be some other type of correlation. So on the network, you can start with just edges and nodes, but you can also add direction to the edges. You can add more than one type of edges. You can add more than one type of nodes, and you can also add information on top of the nodes. And in this lecture, we will see how adding all these information allow us to visualize or analyze some of the biological problems. So one example that we saw implicitly when we talk about single cell data is when we have this kind of a sample by, let's say, features are genes, so gene expression. When we have this type of data from our omics, one thing that we can draw is the similarity between samples. So some samples have similar gene expression profile, some samples have similar gene expression profile. And when we visualize single cell data, we want to visualize each cell as a dot and show them on the screen. But hidden underneath is the similarity score between cells. If two cells have similar gene expression profile, then you can imagine that there is an edge between them, connecting them. And this edge might have the correlation score to represent that these two cells in gray, these are similar to each other in the sense of gene expression. And cells in gray and cells in red might also share some similarity in gene expression, but the cells in yellow might differ from everything else. So underneath our visualization, you can actually draw the similarity, and the edge, in this case, the thickness of the edge, can capture the extent of similarity between the sample. Another type of network that we saw several times are the regulatory network. When we draw arrow from one gene to another or one protein to another, this implies some regulatory relationship. One gene might activate the other gene in a pathway, or it might replace the other gene in the pathway. So these are networks that we frequently saw in biology classroom. In real life, we also saw several networks. When we connect computers together, that's one node is a computer, and the edges are the network connection. So these are also network. When we think about city and street, we have different cities and we have streets connecting them. That's also a network. When we think about internet, we have different websites, different web pages, and they can be linked to each other when you click them. Those are also networks, one type of network. If you think about publication, when people publish together, one node may be a scientist, the age being that they publish the same paper together. You can also create a network of collaboration. Or friendship network, on social media. You can draw connection between people who are friends, and you can analyze whether there are people who are friends with each other as a cluster or as a group. And even with kind of the river and sewage system, we have flows of water from one source to the next. You can think of each source of water like lake and the sea as nodes, and you have river that connects them as edges. So we can use networks to represent diverse systems, diverse scenarios in real life. In biology, when we make networks, usually it comes from omics data, because when you create network, if you notice, one thing that we need to create a network is we need to have a lot of nodes and a lot of edges. Otherwise, the diagram like this doesn't really allow you to analyze anything, if it's too small. So in biology, the network that we are going to analyze or network that we're going to see are usually based on omics data, because with omics data you have all the genes, all the protein, all the mutation, and you have the connection. So for example, in the past, when we want to understand if two proteins bind to each other, we want to know if they can bind to each other, people use the yeast-2 hybrid system, where you attach one protein to the with the DNA binding domain, and to attach the other protein with the enzymatic domain. The only way that you can activate the reporter gene is when these two proteins in brown and in blue, if they can bind to each other, one has the DNA binding domain, so it will recruit the complex to the DNA, and the other one has this enzymatic domain that can activate the transcription. So only when these two can bind to each other that you have the expression of the reporter. If you use two proteins that don't interact with each other, one will bind to the DNA, but the other one will float around. So you don't have the activation. So people use this technique. If they suspect that two proteins can bind to each other, they will attach one to this and one to the enzymatic domain, and then they will see if they can induce the activation. So that's the very old technique. People also try to use mass spectrometry to understand if two proteins can bind to each other. So if you pull down with antibody, one protein, you design antibody that is specific for one protein. When you pull down that protein, if that protein doesn't bind to anything, you expect your pull down result to be purely just that protein. But if your pull down result, your immunoprecipitation result, also contain other proteins, then you suspect that those proteins might be able to bind to your target protein, because you can pull them out from sample with the antibody for your target protein. When we run chip-seq analysis, the transcription factor binding to the DNA, once we know the location of the binding, we can create a network where one type of node is the transcription factor, the other type of nodes are the gene, and we create a connection pointing from transcription factor to the gene. If we think that the transcription factor is binding somewhere near the promoter or the enhancer for the gene, so we have the relationship between transcription factor and gene in terms of regulation. When you have a transcriptomic data from RNA-seq, you can create a gene-to-gene network, and the network you draw an edge if the two genes have the same expression level throughout your condition. Genes that are upregulated together or genes that are downregulated together. You want to draw an edge between them to show that these are related in terms of their expression level being linked to each other. And when we talk about functional annotation, when we want to understand which function or pathway is enriched in the differentially expressed gene, there are many terms. Biological process is the very broad term. All the genes, almost all the genes participate in some biological process. But there are also more specific terms cellular process, or photosynthesis. Like these are more specific processes. So you can draw edges between different annotations, and you draw them if they are directly related to each other. Like cellular process is part of a biological process. Photosynthesis is part of a cellular process. Photosynthesis is part of a metabolic process. So we can draw edges connecting functional terms. And when we visualize, like in this case, we visualize the functional term by first the size of the node. So we now see that the node doesn't have to be the same size, like doesn't have to be even the same color. So in this case, the node size represents the number of genes that are linked or annotated with that function. So you can see how broad or how specific each term is based on just looking at this. We know that biological process is very broad. Cellular process is very broad, but photosynthesis is specific. Translation is specific. Lipid metabolic process is specific. And from your gene expression data, we can also color the nodes based on the p-value. So now you can look at this and say that, I have a significant enrichment of some of these specific process and some of these broad process. So with a single network, you can show both the specificity of the term and also the significance of the term. So it allows you to present more than one dimension of the data. But if you kind of use too much, like if the edges have color as well, then you might kind of be confused like with too much information in the network. So there is like a limit of how much information you can squeeze in. But this is a good visualization type that allows you to put in more than one information into. Another type of network that you might encounter, especially if you are dealing with compound, like molecular compound or drugs or small molecules. So people want to know the effect of small molecules. When we synthesize a new drug, like we want to know what's the effect it would be. So people in this connectivity map project, they use a lot of cell lines, different cell lines, and they treat the cell lines with different compounds. And once they do that, they collect the transcriptomic data. So this connectivity map map the effect of a compound to the transcriptomic data. So if you have a new compound and you treat it on some cell type, you get the transcriptomic data, you can compare your transcriptomic profiles against the expression data in this connectivity map. And if your cells happen to be similar to some of the cells in here, you can ask which perturbation, which compound were used to generate those transcriptomic data. And you can infer that maybe your new drug have the same effect, have the same usage, the same utilization as the drug that they produce in their database. So this is also like a kind of network that can map from drugs or small compound to the cell lines and to the transcriptomic profile. Another example, so this one is a network between viral protein and host protein. So we want to know when virus enters human cells, like which protein from the virus interact with which protein in our body. So in this case, the viral protein are in green, like we color them in green to separate them from human genes. And the human genes, human proteins are in blue. And the genes that are linked to the infection phenotype, like the one that we think are responsible for the phenotype, circled in red. So we can have multiple coloring style to designate different type of nodes and different type of information. And the edges here, some are between the virus and the host, like the one in blue. These are interaction between viral protein and host protein. And there are also edges in black that are interaction among the host protein themselves. In this case, the choice of color might not be too great because it's difficult to see black from blue. But you can color the edges differently as well. Okay. So those are some examples of the networks, of the visualization that we can create with networks to represent biological data. So let's see, in biology, these networks are formed by nature. proteins evolve, and then some of them can bind to each other, some of them regulate with each other. These are caused by the process of nature. So there might be some property to it. Like these networks might have some specific like property, some specific connection. So people about 20 years ago, I think they start to wonder, like once we have all these network data from biology, from real life, like we were wondering, is there any specific structure inside these networks that makes them perform the task, like underlie the organisms or the way we build our city, like the way we build our transportation? Do they have any specific property? And people found that there are several properties that occur in both human-made, man-made kind of networks and also biological networks. And the first property is what people call scale-free. So they call it scale-free because the idea is when you look at these like networks, whether you look at the network as a whole or whether you zoom in like on a certain part, there is one structure that they found to be consistent. So it's kind of scale-free. Like no matter how you zoom, you see the same property. And that property is related to the number of edges that are connected to a node. So if you look at some of these nodes, like this one has a lot of edges, like, but some of them has only few. Like this one only has one, but this one has like a dozen of edges. So if we count like the number of nodes that have small number of edges, like this guy, and the number of nodes that have a lot of edges, and we plot them as a scatter plot, with the x-axis being the number of edges, and the y-axis being the proportion of the nodes. What people found on the log scale, this is the log number of edges, and this is the log percentage of all nodes. And they found that this usually show a straight line. So this is a property where you have more nodes, like with very small number of edges, and you have a few nodes with so many edges. Like these have 100 to 1,000 edges. And no matter how you zoom into your data, when you plot this kind of scatter plot of the number of edges versus the fraction of nodes, you're going to always see the same linear function. And this is called a scale-free property. So this property might not make sense much, like what's the usefulness of being able to zoom in and then see the same type of structure. The second one might be easier to interpret. What people found in real-world networks and biology is the property called small world. So small world means that if you want to go from one place, like imagine if this is a transportation network, like if you want to go from one city to another city, what's the property that you want, like from the transportation network? The property that you want is that you can go from any one city to any other city without having to change the bus or the train like several times. You don't want to change the train 10 times to go from one city to another. You want to change maybe 1 or 2 times. So the small world property is a property that in real world networks and in biological networks, if you want to go from one protein, like one node, to another protein, either via protein-protein interaction or via co-expression or via editing, what people found is that you only need a few nodes in between to connect them. In this case, you can go from here to here with only just 1, 2, 3, 4. You don't need to go all the way around. You just need to go 1, 2, 3, 4, and then you reach the other. So that's called the small world property. And if you search this term, one of the famous examples that people found is that if you pick 2 random people in the world, like anywhere in the world, you pick one person maybe in this room, you pick another person like in the middle of the jungle, like somewhere in the world. What they found is that you can find your friends who know someone, until you get to reach the other person within about 6 or 7 people. So you don't need like to go to 20 people to connect you to some random person in the world. You need only about 6 or 7. And that's a property that we saw both in like man-made networks. We saw in the kind of friendship network, we saw that in the transportation networks, but we also saw this in the protein interaction network, in the gene interaction networks. And that's kind of, this property is linked to this kind of scale-free property somewhat, because the existence of these hubs, like people call these hubs because these are the gene, these are the protein, or these are the person, or web page that have a lot of connectivity. So if you want to send signal, let's say you are inside a biological system, you want to send a signal, maybe this protein is a receptor, and this protein sends a certain chemical outside of the cells, and you need to send that signal to the relevant pathway, relevant functional pathway inside the cell. So the existence of these hub protein or hub gene that can interact or communicate with a lot of other protein and genes will help the signal spread faster. So some example of these are transcription factor. When you have one transcription factor, it can bind to multiple locations on the genome. So one transcription factor can activate a whole bunch of genes, which can then activate a whole bunch of genes kind of further downstream. So people found that this property that allows the cells or the human to send signal from one node, from one protein to another via these hubs that has a lot of connectivities. This also creates the small world property because you can reach one protein to another by going through the hubs, and then you have a short distance between the two. So in real world example, like this in biology, like this hub might be transcription factor. But in real world, like we might think of this hub as major city. Different airlines want to stop in major city, and you can change the airline to a different one. Or if you think about like social media, if you have a famous influencer, that person can spread information or can recognize many other people on the network. So you have these people, these proteins, these airports as a shortcut to send signal or to go from one node to the other. And once people found this property, they were wondering what kind of mechanisms that give rise to such properties in the real world network. And for those of you who are interested, this is a key study about 15, 20 years ago. So they found that there is one explanation why we have these kind of small world scale-free property in many networks. We saw this property in almost every network in real life. So we were wondering if there is any common mechanisms that explain how they form. And people found that there's a simple explanation. When this network grow over time, so one way we can think about this network is that they grow over time. If we think about biological networks, in bacteria, when we first have simple life form, we have only few proteins. So the network is small. But once we develop eukaryote, the animal, and then into human, we have more and more genes, more and more protein. So you can think of the biological network as growing over time. The same way as transportation network, you have more cities, you have more routes adding to the network over time. If you think about the internet, we have more web pages, we have more users, we have more connection over time. So people were asking, how can we explain the property of the network by examining how the new protein, how the new city, how the new users connect to the existing network. When we have new protein, how do they connect to the existing protein-protein interaction networks? And what they found is called a preferential attachment explanation. So if we assume that the new protein, the new users, like the new city, if they have some preference to be connected to the hubs, to the protein or city that already have a lot of connection, if the new node prefer to attach to this, over time, this growth of the network, according to this principle, will create network with these two properties, with the scale-free and the small world property. So we might wonder, like, then why should this mechanism can appear in real life? So if we think about transportation, if you create a new bus company, you want to have your bus connect to the major city. You don't want to operate in kind of rural area. You want to operate and have at least one connection with the major city. If you are a new user in the internet, you are likely to follow someone already famous. So you have some tendency to connect to hubs. In biology, they don't have a clear explanation, but what we can imagine is, for example, like if we think that a transcription factor, which is already a hub, like it can bind to multiple locations on the genome. So when you have new genes, like it appears somewhere on the genome. So there are chances that it will appear near certain transcription factor binding site, and that transcription factor binding site might facilitate the transcription for that gene when they emerge. So this can explain somewhat that when you have new genes adding to the genome, it might join an existing regulation of the existing transcription factor. So you can have new protein or new gene being more likely to be regulated by the existing hubs, which are the transcription factor or any other regulators. And for real-world network, it's easier to imagine. You want to connect to major city, you want to connect to famous people. And the consequence of this preference that the new entities, new protein, wants to connect to an existing hub is the Disu property. If you're curious, like how does this mechanisms derive or arrive at Disu property, they have a computational proof of the concept. But for this class, we will just explore that this explanation is a plausible explanation for why real-life networks and biological networks have these two nice properties. Okay, so these two properties are not the only two properties that people care about. Okay? So in biology and in mathematics, people want to measure other properties of the networks. And some of these will have implication and application for us as well. So let's look at some of them. So why do we want to care about the connectivity in the network? So when we draw network diagram, like whether as protein as node and protein interaction as edges, one thing that we imagine that the networks are doing is that they must have transmit signal like from one node to the other. When signaling pathway operate, like the receptor takes signal from the molecules outside the cell, and it phosphorylates maybe the next protein, and then activate another protein, and then activate another protein. Or when protein bind to each other, it can form complex that can activate different functions, different pathways. So we think of networks in biology as constantly sending some signal from one protein to the next. So the connection is important. Another way to imagine that the connection is important is when we have disease or when we have perturbation. When you treat the cell with certain drug, we imagine that the drug and the disease might disrupt some of the protein. Or when you knock out the gene, you essentially remove one of the nodes from the network. And when you remove a node or when you disable a node, the function of the node, you are essentially changing the connectivity inside the network. So people can think of visualizing the impact of a disease by showing you that if you remove this node, like in the middle, how does it impact the connectivity of the other protein? Versus if you remove this node 14, if you remove it, like almost nothing changed. The protein 13 and 15 might have a problem sending signal to each other, but they can still reach to the other protein. But if you cut node 12, like node 10, you are disrupting the flow of signal between the two halves of the network. So this type of perturbation idea can also be visualized in terms of the connectivity of the network. So we have some concepts about how important nodes and edges may be. If we have nodes that are connected to many nodes, protein that interact with many proteins, transcription factor that regulate many genes. Those might be important. So when we think about biology, important might mean it's highly connected. Nodes that are linked to many other nodes. When we think about importance in terms of signaling, in a signaling pathway, we might think of important genes as nodes that connect other genes. By itself, it might not connect to many genes, but if two genes want to send the signal to each other, it has to come through here. That can be another definition of importance in terms of signaling. Another importance might be in terms of disease. Like if you disrupt a gene, if you knock out a gene, which one in this network would have the most impact on the overall network? So that can be another definition of importance. Which genes are located at the center of the network? So by looking at gene and protein and looking at their interaction, their relationship via networks, we can think of many ways to identify important genes and many ways to define important protein. So let's see how we can calculate some of these scores. Like if we want to rank the gene based on how highly connected they are, like how should we calculate? If we want to show that some genes are involved in connectivity, like in signaling between other genes, how should we calculate that? If we want to show that a gene is at the center of the network, how should we calculate that? So when you read or when you use tools that analyze network data, these are the keywords that you see frequently. So a degree is the number of edges that are connected to a node. So if I ask you like, what's the degree of D here, you will count 1, 2, 3, 4, 5. So the degree would be 5. So the degree kind of represent the connectivity of the network, of the node. Locally, this node D is connected to 5 other nodes. If I ask you about clustering coefficient, so this is clustering coefficient is the proportion of neighbors of the node. Okay, so the clustering coefficient, the idea is if we want a clustering coefficient of a node, like let's say node number 2, we will examine the neighbors of node number 2, which are 1, 3, and 3. Okay. So there are 3 nodes. And what we want to ask is how many edges are there among the neighbors of node number 2. So between 1, 3, and 3, let's say if we assume that there is no edge between 1 and 3 here, the dashed line. So we have 1 and 3 being connected, 3 and 3 being connected, but not 1 and 3. So we have 2 edges. And the total possible edges between them is 3. You can only have 1, 2, 3 edges. So in this case, the clustering coefficient is the proportion of the node that you found versus of the edges that you found versus all the possible edges. So why is this interesting? So the clustering coefficient tells you not whether node number 2 is connected, but whether the neighbors of 2 are connected. So if you have a node with high clustering coefficient, meaning that all its neighbors are also connected. That means that if you remove node 2, if you remove it, if you either knock it out or the disease can disable this gene, the other genes can still communicate because you have high connectivity among themselves. But if you have a node with low clustering coefficient, for example, this node D, it has 5 neighbors, and among 5 neighbors, only 2 of them are connected. So if you disrupt node D, either by disease or by drug or by anything, you are destroying the connection between these 5 nodes. So clustering coefficient can be a measure of how this node will respond to the perturbation, to the disruption. High cousin coefficient might mean it's resistant to perturbation because the other node can still operate. If it has a low cousin coefficient, if you disrupt this, you disrupt the connection between all the other nodes. So it might be sensitive to treatment or sensitive to disruption. Another concept that we'll talk about is the idea of a path. If you want to send signal from node A to node B, it has to go through some other gene, like other node. And what you can calculate is the length of this path. In this case, it uses 1, 2, 3 edges. So the length of this, the distance is 3. So how far, like if you want to send signal from one gene to the next, to the other genes, like the distance between them can tell you how efficient or inefficient the signal can flow through the network. So for example, we already talked about some of these. If you have a node with high degree, right, so these nodes are hubs that interact with many partners. So this might be important. Regulator might be important genes. If you have a node with high cousin coefficient, so the neighbors are also connected. So if you disrupt the center node, it might be resistant or resilient to the perturbation because the other node can still communicate with each other. In terms of signaling path, like if you have a short length, that means you can send signal from one gene to the next with few genes in between. But if you have a long path, that means the signal has to go through multiple protein, multiple genes to reach the other. And in computer science, there are ways that if you create a network of protein and genes, and then maybe have a binding constant or maybe have a co-expression score, you can create the shortest path from one node to the next by different algorithms. And in this case, the one that people use a lot is called DISA algorithm. I want to refer to this because it's also used in dynamic programming approach. And we already saw many examples of dynamic programming. The idea is if you have a network with 8 nodes, this is your complex problem. But if you remove one node, or if you start with a certain position and you start to record only the shortest path, then you can keep doing this, and you will eventually fill the path length throughout the network. So it's an iterative, it's a dynamic programming approach. Okay, but that's not important. Just want to let you know that we have dynamic programming to calculate these properties, even though it seems to be complex. Okay. What I want to highlight is there are many ways that you can view network data. As a biologist, like you might view it as protein and gene, right? And the connection might be binding, might be regulation. But when you describe protein and gene interaction as binding or as regulation, we don't know the strength of regulation. You cannot put numbers. Like if one transcription factor activates the other genes, like what is the interaction or what is the regulation score? We don't really have way to quantify that in biology. So people from engineering, they step in and they say that if you don't know how to model or if you don't know how to calculate interaction strength or association score in terms of biology, why don't we transform it to real world system where we can make sense? For example, some engineering people might view biological network as a system of flow, like water. You might have different genes being different reservoir, like different lake. And the binding between genes or the signaling pathway can be thought of as rivers that connect one reservoir to another. And if you want to measure if you have a receptor protein here, and there is a signal, maybe from some molecules, like from nutrients coming in. When you imagine this as a reservoir and rivers, like the signal will become the amount of water. So let's say you have an equivalent of 1 million liters of water being poured into this node, into this lake. The question that engineering people can simulate, the problem that they can solve is, if you have these lakes that are connect to each other with rivers that have a certain capacity, after a certain time pass, how much water, starting from 1 million here, how much water will be in different lakes. So you can imagine this as a receptor getting a nutrient signal and then sending a signal to different proteins in the network. And we can calculate how much the cell will respond based on different protein, different pathway, based on how much signal can flow to the other protein. So engineering people view this as the flow of water to the network. But if you ask an electrical engineer, they will view your gene and gene-gene interaction as a circuit. So if two genes can bind to each other, if two proteins can bind to each other easily, it's like you have a low resistance. But if two genes don't interact with each other, it's like you have a high resistance. So once you draw this diagram out with the joint, the nodes here, like being the genes, and the connection here being how well they can bind to each other, you can have a circuit. And if you think that this is the receptor protein, this receives the signal, so you apply a voltage as a circuit. You apply a voltage between 2 points A and B, and what electrical engineer can calculate for you is how much current will flow to different parts of the circuit. And any part of the circuit that has high current, like high electrical current, that is the signaling part that are important. Because if you want to send signal from this protein to this protein, it has to flow to these proteins. These are the ones with high electrical current. So in this way, engineering idea can transform abstract biological interaction into something that you can imagine, something that you can solve. But whether it's correct is another matter. Whether we can view protein interaction as scaling in the same way as resistance is a question. Whether it will scale as a flow of water is a question. But at least there are computational ways that can quantify how important a protein is, how important a connection is, based on these signals. Interestingly, if you ask a civil engineer, they will come up with a different way to view the biological networks. So for example, they might view different genes, different proteins as seedings, and the interaction between gene and protein as the stream, as the transportation that connects them. If two proteins can bind to each other easily, with strong binding, you have a highway between them. But if two proteins bind to each other very weakly, you have a narrow array to connect between them. And if you want to know how the signal flow inside this network, since we model them as city and street, we can imagine people living on this network. And people can travel to the street, from one city to the next. So this is a similar idea as random walk. So we have many particles, many people, many persons in this network. And each one of them can travel to the adjacent city with certain property, with certain probability. And we will keep running this for maybe hundreds or thousands of people in the network. And at the end, we will ask, what's the density or what's the frequency that people go from one city to this street? How often is this street used? And from this visualization, you can simplify the structure of the network into just 4 components. One component here, one component here, one here, and one here, based on how frequent people travel inside the network. So different view from different disciplines can help making sense computationally of the structure of the biological network. In terms of mathematics, so another different way, we talk about degree, we count the number of edges that connect to a node. But the others, there are so many ways that people in mathematics can define the structure or the connectivities inside the network. We are not going to worry about the formula here, but we are going to enjoy the what being captured by these different definitions. So if we think about degree, like degree is the number of edges. So nodes with high degrees are here, like with deep red color. And you can see that degree is a very local score. Like node with high degree here, there can be node with high degree everywhere in the network. It's not a global property, it's a very local property. You only ask how many edges do I have. But betweenness, which is it tells you if a node has high betweenness, this means that if you want to send signal from one node to another, it usually has to go through this node. If you have a node with low betweenness, this means that when you send signal from one node to the other, it doesn't need to go to that. So you can see that nodes on the periphery of the network will have low betweenness because when you send signal, it doesn't have to go through the periphery. It has to go to the center. So betweenness is a global score. It tells you which node is important in terms of signaling, in terms of sending signal from one node to the other. And you can see that there are few, like compared to degree, like you can have many nodes with high degree. But for betweenness, only few specific nodes can have high degree because these are the nodes that connect everything on the global scale. Closeness score, this is similar to the concept of center. So you see that closeness score and harmonic score, these are high in the middle of the network and low on the periphery. So it's different from degree, like degree can be anywhere depending on how many edges. Betweenness is in the middle, but not everything in the middle, only the ones that are essential in connecting the flow of signal. Closeness gives you everything in the middle. So different kind of formulation give you different concepts of the properties inside the network. The one that biologists want to use are usually degree, betweenness, and closeness, the one on top here, because they can interpret degree being having many connections, like these are hub gene or hub protein. Betweenness are the ones that are important for signaling, and closeness might be the ones that are important in terms of disease, because if you have disruption of node with high closeness, the impact can spread to broad area of the network. So for example, like if you look at this example, this node H and I, you can imagine that this node H and I are probably the one with high betweenness, because H and I connect this half of the network to this half of the network. So the term betweenness, like you can remember easily. These have high betweenness because they are in between something. They are H and I are between this part of the network and the right part of the network. So they are between something. They have high betweenness. This node P has high closeness because from node P, it can reach other node with the shortest distance. So closeness means it's at the center of the network. From this visualization, it doesn't suggest strongly that P has the highest it's the center of the network, because it doesn't look like it has the center. So closeness centrality is sometimes difficult to see, because from the structure, we can see H and I easily as a bridge between two halves of the network, and we can see node J having the highest degree because we can count. But for closeness, it has to be the one that can connect to everything with the shortest distance, which sometimes can be difficult to see. And in biology, we might think that multiple definitions of importance, like high degree might be important, high betweenness might be important. We might mention that maybe these properties should point us to the same gene. because we have certain genes that are important. Should they have both properties? And it turns out in biology, different genes can have different importance. So for example, let's imagine this example. If we have two functional pathways, two different pathways that are communicating with each other. So we have many proteins in one pathway, like these are connected to each other because they're in the same pathway, they are working together. The other protein in a different pathway, like these are also working together. And there are only a few proteins that communicate between them, sending signal between them. So in terms of connectivity, in terms of degree, the node with high degree would be the one in the pathway, in the middle of the pathway, will have high degree. But the node with high betweenness will be the one that connect across pathway. And node that connects across pathway usually have a low degree. So in early study in biology, people found that these different kind of score, like different criteria for importance in the network, doesn't necessarily occur in the same gene. You can have gene with importance in terms of betweenness, in terms of signaling, but low importance in terms of regulator. You can have gene and protein that has some importance in terms of potential regulator, but it doesn't send signal across the pathway. So these are kind of what people found when people start to analyze biological networks about 15 years ago. So when we have networks, in addition to calculate the score, like calculate the connectivity, we can also do other things. One thing that we have done before, we have looked at quickly before, is how we can cluster cells, cluster genes based on the network. In single cell data, we have the node being the single cells, and we have the connectivity between the cells representing the similarity in gene expression profile. And back then we talked about modularity score. If we want to have a good cluster of cells, like into cell type, we want the cluster so that the number of edges within the cluster, like the number of edges that connect node with the same cluster with the same color, we want them to be high, and we don't want many edges that connect across different colors. So the top here, you can see that it's a good clustering result. The bottom here is a bad clustering result. One thing that I want to add here that I didn't mention back then is a concept called resolution. And that appears when you visualize this as network data. And if I present to you these two results, the clustering on the left with two clusters, blue and red, and the clustering on the right with 5 clusters, which one is better? Both of them looks correct, right? The network clustering result on the left have 2 clusters that each are connected. But on the right, right, we observe that in the blue cluster, there are actually 3 nodes that are connected and 4 nodes that are connected. In the red clusters, there are 3 nodes that are connected, 4 nodes that are connected, and then 5 nodes that are connected. So depending on how detailed, how deep, how high resolution you want to go, when you have network data, you can create clusters at different resolution or different depth. And there's no one to tell you which one is correct. So the only way, if this is a single cell gene expression data, the only way to tell which one is better would be to identify the gene marker for the orange group, for the green group, for the cyan group, for the yellow group, for the purple group, and see if it matches to any known cell type. And that can tell you that the clustering at this level might be better because I can identify a different type of macrophage, for example. But versus on the right, on the left, you might identify multiple types of macrophage as a single cluster. We saw examples like this before. Okay, because I was late, so I'm going to skip some of these. So when we perform network clustering, I haven't told you the name of the algorithms. So nowadays, when we want to cluster networks, we usually use either Loven or Redden algorithms. So these two operate in the same way. They want to maximize the modularity score that we have seen before. And just to show you, this is from a research that want to analyze the position on the DNA that in 3 dimensions fold into each other. So they want to know if certain location on the DNA, do they fold and meet each other in 3D, like as a group. Like you might have multiple genes being folded in 3D to form some kind of a network in 3 dimensions. So in this case, we're going to learn about this data in maybe 1 or 2 lectures. But basically, you can create a network that connects different loci on the DNA to each other based on 3D distance. And from this network, this is visualized as a heat map. So each rows and each column is one loci on the DNA. And the colors means that whether they are near each other or far away from each other. So with network clustering, we can identify certain position from chromosome 1, 2, and 3 that are connected, that are near each other in 3 dimensions. So there's something going on inside the nucleus, that some part of chromosome 1, some parts of chromosome 2, some parts of chromosome 3, even though they are on a different chromosome, but inside the nucleus, they are located, like genes in this area are located near each other. In the next lecture, we'll talk about the importance of this phenomenon. But for now, this is another example of how we can cluster data in the form of networks to understand biology. Another way that people look at networks is they want to look for motifs. So we heard this term before. Like when we think about transcription factor binding to the DNA, to the genome, we know that transcription factor must have recognized certain DNA pattern on the chromosome, and we call those motifs. Those are patterns that transcription factor can bind to. So motif means something along the line of patterns that you see many times. So in the case of network, there are multiple types of patterns that you can expect to see. For example, regulatory patterns, like you have multiple transcription factors co-regulating the same gene. So you can have signal amplify each other. You can have a cascade of signaling. One protein activates the next, one activates the next, one activates the next. These are recurring patterns inside biology because these allow the cells to amplify the signal better. So the cells, the evolution, we use this pattern a lot. Another example, so we learn about protein-protein interaction, like when two proteins bind, you have protein interaction, physical binding. Another interaction type that we might not heard about much is called genetic interaction, or another name is called synthetic lethality. So this genetic interaction means that you have two genes. When you knock out one gene, the organism is okay. When you knock out the other gene, the organism is okay. But when you knock out both genes together, you have a lethality, the cells die. So this genetic interaction tells us something interesting. You can disrupt one gene, the cell can resist that. You can disrupt another gene, the cell can resist that. But if you knock out two genes at once, the cell die. So this means that these two genes must have some function that they can operate in place of each other. But if you lose both of them, then there is no backup. The cells cannot survive without any gene that perform that function. So people found that this genetic interaction tends to be linked to physical interaction. And the idea is when you have multiple pathways, like multiple signaling pathways, multiple functional pathways, each of them, each pathway usually communicate via protein-protein binding. So inside each pathway, you have physical binding as a means of sending signal. So when you knock out protein right, from one pathway, the cell is still fine because most of the pathway is still intact. But if you knock out one protein from one pathway and you knock out another protein from a different pathway, now the cells have two defects. And in many cases, in simple organisms like yeast, when the cells have two major defects like in two different important pathways, it cannot survive. So people found that if you have important pathways, like two important pathways, the protein that are in different pathways, these tend to have genetic interaction. You cannot knock out both of the genes. The cell will not survive. So people found that the genetic interaction and physical binding of protein interaction tends to complement each other. You have protein being connected with physical binding and protein being connected with genetic interaction in an orthogonal pattern. So this leads to an interesting approach that, so if we expect that in simple organisms, like if we have protein interaction in one group of protein and interaction in another group of protein, then we expect genetic interaction between them. So what happened if we look at the current knowledge and we found that between A, B, and C, like these are from one pathway, A prime, B prime, C prime, these are from a different pathway. And we only know genetic interaction between them, like the one in red, are genetic interaction. But we don't see this, the one in gray. The genetic interaction between B and B prime haven't been observed. Okay. Or in this case, we have 4 proteins, and we already have genetic interaction between 2 proteins in the pathway. But we haven't saw this interaction, we haven't seen this between D and B prime. So people come up with an idea that maybe there are genetic interaction, we just haven't tested yet. Because we know that we don't have a complete information about the interaction between every pair of genes. Like there are too many to test. Like, so people found that if we have an almost perfect pattern, but we lack some of the edges, some of the interaction, maybe these ones are correct, but we haven't known it yet. So this might be candidate for experiment. You might want to do follow-up experiment to confirm synthetic lethality or genetic interaction between B and B prime or between D and D prime. So by analyzing the network, by looking at these patterns, these motifs that we expect to see based on biology, but they are not complete, they are missing something. We expect that the missing data might be because of our lack of knowledge, and this can create new knowledge. So the question is, for this kind of motif, like this pattern, how much can we trust that the missing edge here are real? They are real but missing because of our lack of knowledge, and not because in biology there is no interaction here. What are the chances that we can trust this kind of approach? So people want to be able to show how much confidence, how much p-value that we can expect to see from this pattern. So this takes us back to, I think, I forgot which lecture, but we talked a little bit about if we create a network and we want to test if we observe some property in this network. Is it a true property of the network, or is it something that you can see in any network? Someone can do a random network and you will see the same property. Back then we showed that using permutation tests, we can generate many different random networks that have the same number of nodes, the same number of edges, but different connectivity. And then we can ask, do we see the same motif? Do we see this pattern? One group of protein being connected with protein interaction, and then they are connected with genetic interaction. Do we see this if we shuffle the data, if we permute, if we randomize our biological network? So people, since we need to generate random networks to ask if we can still see the motif, there are many ways that we can produce the random network. So in the past, people first start with what's called a completely random network. So you generate the node, like you know the number of genes, you know the number of edges, but the way to connect them, like connect the edges to the nodes, we do this randomly with certain probability. People found that this is not a good way to generate a network because you can see that there are many nodes that are not connected to anything. So this kind of randomizing the network, even though it's random, it doesn't give you something that resembles biological networks. In biology, we don't have these singletons. So people start to refine the idea that maybe can we add some structure to the network, and we still draw the edge randomly, but we draw it randomly under some constraint. Can we create networks that are more similar to biology? So there are these two approaches that can create something that more resembles the connectivity in biological networks. Okay, so besides motifs, so motifs are patterns that we expect to see in one network. We can extend this idea by asking, can we expect to see similar structure across network? And in this case, we think of network as when we think of network as being growth through evolution, from simple organism to complex organism. So what we can do is we can try to identify the same pathway across different species throughout the evolution. And we want to know how nature evolved this pathway. From the simplest organisms, we might only have 6 proteins in the pathway, and they're not even fully connected or connected. In more complex organisms, we might start to have more protein, more protein that emerge, and they participate in this function. So we start to see more components. And what we want to know is, can we align the structure protein that are related to evolution? Can we see the change in structure of the pathway? In this case, we see the change in structure from the most simplest organism to the one with more protein, to the one that now has a lot of communication, a lot of interaction between the protein. So people can use this to study the evolution of pathway and visualize in terms of network. So this can summarize that concept. We want to know throughout the course of evolution what happens. Is some protein interaction lost? Is some new protein being gained? Is some new interaction being formed? We want to know how the evolution operates on protein network. Okay, so for the last part of this lecture, we're going to go through some application where we can use network visualization or network data to understand biology. So the first example, we mentioned this as perturbation analysis. So what we want when we want to understand the importance of each node or each edge inside the network, we can calculate the betweenness score, we can calculate the degree. Those are also good way to understand the importance of the network. But in terms of disease and in terms of perturbation, we want to understand the effect of a node in terms of what happens if we don't have that. What happens when we lose that node? What happens when we lose that interaction? What would be the change in signaling, how the signal flow across the other networks through the nodes? So from this example that we saw before, we have node with high betweenness. We know that if we knock out this node, we no longer have information flow across the two parts of the network. So this might have a strong response to perturbation. Versus if you knock out this with high closeness, it's at the center. But in this case, even though it's at the center of the network in terms of the distance, in terms of the structure, if you knock out P, you don't expect much to change in terms of the communication inside the network. because P is kind of outside of the main connection. So this the way that we analyze the perturbation kind of give us a different interpretation compared to the score that we can calculate on the network. Another technique that we can do is we can integrate multiple types of networks. For example, if you integrate drug and gene network, you know which drug affect which gene. You have one type of network. You have a network between gene and disease, which gene cause which disease. You have another type of networks. If you connect them together, what you have is a way that you can start with a drug. You know that this drug affects these two genes. And the disease that you want to target is this disease, and they are known to be caused by these three genes in red. So if you want to know if your drug might be effective against this disease, one way to visualize or to calculate on the network is how far away from your drug target, how far away it is from all these disease-related genes. So you can combine networks and calculate the distance on the network to connect from your drug to genes, from genes to gene, from genes to disease to get the interpretation. Another important way to use the network, similar to how you kind of propagate from drug to gene to the disease, is for mutation data. So the thing is, like when you analyze cancer, if you study cancer, you will know that no two patients have the same mutation profile. It's impossible. We all have different mutations. So if you want to classify patients based on mutation profile, it's almost impossible because there is no similarity between them. But we can imagine that if we have a mutation on a node, like on a gene, we can imagine that if that gene regulates another gene, or if that gene interacts with another gene, the impact of the mutation should spread like from the original gene to nearby gene as well. So we can imagine that the area that are affected by the mutation is not just a single gene, not a single dot on the network. It's an area inside the network. So people use this concept when we have two patients with different genotype, one in yellow, one in blue, mutate at different genes. But this gene might be in the same pathway. So if we propagate, if we let the effect flow from the mutated genes, like imagine as you pouring water into the node and let it flow inside the network. What we want to see is if we put in yellow color water in one node, and we put in blue colored water in other nodes, starting from this node for each patient. Where do we see the mixing of the two colors? Where yellow meet blue and create a green color. So this kind of a propagation-based analysis start with perturbation or mutation at certain point in the network, but we let the effect flow and propagate inside. And we want to identify if these two patients are similar or not, depend on how much common area that are affected in both patients. And in this case, we can identify this part of the network where the blue dots and the yellow dots run the signal kind of run into each other, and we see a lot of green colors here. So this, based on this analysis, we can say that these two patients, even though they have totally different mutation profile, this pathway is where they are both affected. So we can find some similarity between them compared to before. Okay. And the last example, I want to go back to the differential expression analysis. When we analyze transcriptomic data, we perform differential expression. We compare the identified genes that are up or down regulated. And then we have one functional enrichment techniques where we say that we want to see if our differentially expressed genes are they connected on the protein interaction path network. We want to see our differentially expressed genes, whether they are on the same area, on the same pathway inside the network. In that analysis workflow, we start from differential equation without looking at the network, and then we map the genes onto the network. So some people propose, right, why don't we do differential expression together with the network data? So in this case, we are going to compare both gene expression profile from two conditions. So these are samples from maybe treatment and control and different genes. Each gene, we know the location on the network. So based on the fold change, based on the expression difference between the two conditions, you can color the nodes based on the fold change. And with this algorithm, we want to identify region on the networks, connected region on the networks that have similar or significant fold change. So this kind of integrate differential expression and functional enrichment using the network data together into one step. We identify differentially expressed subnetwork, so part of the network that are differentially expressed. So we already conditioned that if you want to call something as differentially expressed, it has to have different gene expression, and also it has to form a structure on the network. So we can combine the two techniques together at once. Okay, so far we have 3 lectures on the module of system biology, where we recognize that cells and disease, these are systems. They have multiple components like affect multiple genes, multiple proteins, and the system can change over time. We look at how we can model change in gene expression with differential equation. We can simulate how cell can change from one state to the other. We look at ideas and benefit that we have from integrating multiple types of omics data together. And today we close with the visualization and analysis tools that are based on networks data, where we can see interaction between genes and proteins, and we can make a lot of interpretation about which gene is important, which interaction is important, what happens if we knock out a gene, what happens if we have a mutation, for example. So from next class, we will go into a new module about structure. So it will be about protein structure, RNA structure, and the structure of the genome, the chromosome. Okay, so that's everything I have for today.